Thank you, Ben. My name is Mark Flynn. I'm a software developer at Los Alamos National Laboratory, and I'm going to be talking to you about KBase taxonomy apps that we've put in as part of our SFA. This is work I've done in collaboration with my colleagues at LANL. Jen Shilo and I were the ones who added these apps to KBase. Paul Lee was the one who developed the Gotcha2 app, the main one that we'll be talking about today. And Patrick Chain is our PI. This was done as part of the bacterial fungal interactions SFA. If you first, I'll tell you about the apps that are in the KBase App Store. Tell you how they work, a brief description of how they work. I'll give you a demo of how I would use these apps in KBase, and then some tips, tricks, things to keep in mind when you're doing taxonomic profiling using these apps in KBase. So if you go to the app catalog and you search for taxonomic, you'll get two apps that come up for taxonomic profiling, Kaiju and Gotcha2. If you try the version, change that to beta, you'll get two more apps, Centrifuge and Kraken 2, that we're now working on getting. They're about ready, but they're not quite ready for release, so they're still in the beta stage. Gotcha2 is the app developed at LANL by Paul Lee. It's a metagenomic taxonomic profiling method. It's gene independent and signature based. And the way it works is that you take, for each given taxonomic level, remove all identical k-mers or words that between organisms at a given taxonomic level. And as you keep going down the taxonomic tree, you keep removing more and more sequences that are in common at that level until you get down to the very lowest level. It's the remaining fraction that's unique for that genome. And now you can use that as a simple FASTA database that can be queried with traditional tools. So now that you've got the signature for your organism we're calling X, you can take some sample reads and map those to the signature. You get 12, in this case, you get 12 hits. Concatenate them, and you can look at the coverage, and in this case, it's 43.9%. Now, Gotcha2 has two different databases, and which one you use depends on the type of sample that you're looking at. One is RefSeq, bacteria, archaea, and viruses, and the other is a fungal database also using RefSeq. So there's far fewer ones in the fungal since there's fewer genomes in RefSeq. So for the other taxonomic profiling apps, the one that was already in the KBase App Store is Kaiju, and that's an amino acid based taxonomic profiler. It converts the reads into amino acids and then queries against a protein database. Centrifuge, it uses the Burroughs-Wheeler transform and Faragina-Manzini index, and it's optimized specifically for metagenomic classification. Uses a fairly small index, which is nice if you're running it on your own outside of KBase. Kraken 2 is a k-mer-based taxonomic profiler. It takes a database it maps the k-mers to a database and finds the least common ancestor of all genomes known to contain a given k-mer. I think somebody else has done this analysis where they compare the different taxonomic profilers, looked at true positives, false negatives, and false positives. You can see from this graph that Gotcha2, Kraken, Kaiju, Kraken and Centrifuge all pretty much come up with the same number of true positives. However, where they really differ is in the number of false positives. Gotcha2 has a very, very low percentage of false positives, only 2 out of, it only had 2 false positives. And Kaiju had, almost 3,000. And I should say these were tested on a synthetic data set. So we knew exactly what species were in that sample so that you can give a true profile of how well they perform. So now I will give you a brief demo, show you about the output, and then I'll talk about the tips for running the KBase apps. Let's see, need to stop sharing. Sorry. I apologize. The sharing part I'm not too familiar with on. Sorry, Ben. Just stop sharing.
It should be at the bottom.
Oh, here it goes.
Yeah.
I apologize.
No worries.
Okay. And now I want to start sharing. Okay. Okay. Oops, sorry. I want to use this one. So what you should be seeing is just a blank narrative with gotcha 2 at the top.
Yep.
Okay, great. Okay, so I will show you briefly how I would go about running the workflow, and then after each step, I'll show you where it's already been run. So the first step is if you've got a bunch of raw reads, you probably need to do some kind of pre-processing to remove low quality reads. So I use Trim-O-Matic. The Trim-O-Matic, you select the sample you want to run. In this case, we're using HMP. You can search in the. So you first off, of course, you have to add the data to your sample, to your narrative. So I'm going to use the HMP meta stagger keys stagger. Okay. And then there's a bunch of options that I will not go into, but there's one thing is nice about I love about KBase is you can go into the it usually has a lot of information about how these different apps work and the different options and things you might want to. Okay, so the only thing you really need to do is change the to give it a name for the output. So you click run, and then we'll click over to the output. So it just tells you, for instance, how many reads were in your original sample, how many are remaining after running through Trim-O-Matic, and then how many were dropped. And then it produces a new object name, which are called HMP Stagger QC. And you'll see that in the output up here. So it adds the use a trim to the output, whatever you name it. Okay, so now we've got our trimmed reads. We will run our first analysis using gotcha 2. Now gotcha is there's only one decision that you have to make, and that is the type of database you want to run, whether it's the viral and bacterial or the fungal. But if you'd like, you can look at the advanced options. So minimum coverage. So there's quite a few things that will help you weed out samples that are or results that don't meet your criterion. For example, minimum coverage. So that how much coverage is necessary. What's the minimum amount of coverage needed before you want to add that to look at the abundance calculation. Then minimum number of reads, the minimum length of the reads, and this is maximum z-score. So this one's probably the one that's not very self-explanatory. This one's really for some more pathological samples or pathological cases where you have, where you have all the reads mapping to one region of your sample of the database. So you've got a lot of redundancy. You have a very high degree of coverage, a lot of redundancy. But it's just for one section of your of the database, and you probably don't want to include that. So you weed those out by using this maximum Z-score. And you click, or you add your reads. So in this case, it's HMP stagger QC. Okay, and then click run. Okay, and then we can look at the output. Okay, so the results page has 3 different tabs. One is the results table that just lists at each level how many with the tax ID, how many reads, the number of base pairs, and a whole bunch of things about how your sample was, what the results from Gotcha2's processing of your sample. So at each level, class, species, and you can go on throughout several pages of these. So you can show at different levels, phylum, order, and so on. So you can also look at a dendrogram of your sample. And so it shows how all the species, all the organisms in your sample were related. And I think the size of this bubble here is the number of reads that were mapped to these taxa. Also, you can see a Chrono Plot, which is, I think, a very, very pretty. So these show how all your reads map as a percentage of the mapped reads in your sample, and at different levels, which taxa were present at each level. And okay. Next, we can try rerunning this with a fungal sample. So if you want to do the fungal sample, you just run do this exact same thing. Click run, and then you look at the output. So for this sample, it says error. I mean, I don't know if this is the best way to show this kind of result, but this is actually expected. So if there's no taxonomic classification, so your sample just didn't have any fungal reads, this is the output. Maybe a better way of doing this would just be a non-error, but it ran as expected. But see, it's kind of hard because right now we're having some difficulties with the fungal data with running fungal samples. I tried running some samples that worked before with Gotcha2 fungal database, and for some reason they are not running at the moment. So I would not recommend using trying to run fungal samples. We filed a ticket with KBase and we're working on that now. Okay, so next I will show you Kaiju. Kaiju is the other database, the other tool that was in the KBase App Store. And so KBase, this has Kaiju has some questions you need to answer before you click run. So for instance, you need to say what level you want to run them at. You could pick a taxonomic ID, a taxonomic level, and then you can pick the database. They have 4 choices, depending on your sample, whether it has eukaryotes or not. And then abundance and subsample percent. So I think subsample is if you want to do a quick and dirty analysis, you don't need to know like at the sample level, you can just pick you can do a subsampling to remove some of the reads that are redundant. And then advanced, if you look at the, oops, yeah. Sorry. So this is one of those things I noticed with KBase sometimes is it won't let you scroll down. Okay, there you go, cool. Show advanced. Let me just reload this. One of those strange things that happen sometimes with KBase. There was a question about Kraken 2. All the analyses were done with default parameters. Okay, so let's try Kaiju again. You see I'm looking in my favorites, but if you search in Kaiju, so these are all, they should be in microbial communities. All right, so show advanced. Okay, so they have quite a few parameters that you can play around with that will allow you to filter according to how precise an answer you want, how much you want to filter out unwanted reads. So we will click. And pick all and click run. Okay, now let's look at the output of Kaiju. All right, so they present theirs as these stacked plots, their output. So for each stacked, at each taxonomic level, it has a stacked plot of the percentage of reads that map to a particular tax ID at that level. So class, order, family, and so on down to species. So these all these can be shown as so you can open these as a separate tab, which makes it a little bit easier to read than inside the notebook. Okay. All right, so and then you can also see it as a Chrono Plot. So very nice. And so you can see there's it found quite a few taxa in this sample. Way if you notice a bit more than in. So we can open this also up in a separate tab. So yeah, Kaiju finds quite a few more taxa than Gotcha 2 did. Okay. All right, and then let's look at the two that are not in that have not been released. So we click on the beta to get let's see, let's try Kraken 2 first. It's also in microbial communities. Okay, with Kraken 2, you select your sample. And then the only thing the other select is the database. There are 5 different options. There's a standard Kraken, there's a mini Kraken, and then there's 3 of the standard 16S ribosomal RNA databases. So if your sample consists of 16S RNA, you would probably pick one of these. And then you click run. So Kraken, for instance, and Centrifuge have more options available than we've implemented in the app. So you, if people are interested, we can add those to the app fairly easily. And let's see, then we can look at the output. And we've made the decision to pretty much use a similar output as Gotcha 2 for Kraken 2 and Centrifuge. Somebody asked how do GTDBTK and MOTUs compare to Gotcha. I'm not as familiar with those other taxonomic profilers, so I really can't say what they're like. Can one build a custom reference database for Gotcha 2? Yes, you can. There is code that's been released that you can give it the FASTA files or FASTQ files and build your own Gotcha 2 databases. Our eventual goal is to add that as a capability to KBase, but we have to work out a lot of details for how exactly we would get that, how that would exactly get done, since it would allow the database construction takes a lot of time and a lot of resources. So that's something we need to work out with the KBase admins. Oops. Okay, so we can error loading the results. Okay, here we go. All right, so again, you can look at the a table similar to Gotcha 2, listing the different species at each different level. How many reads are there, and we can also create a dendrogram. So also you can see that there's quite a few more taxa found by Kraken 2, and the Chrono Plot is quite busy. So you might be wondering, okay, is Kraken 2 why would people use Kraken 2 instead of Gotcha? Kraken 2 and Centrifuge are better suited to per reads classification. Gotcha 2 is well suited to per sample reads classification or taxonomic classification. Okay. All right, so next we'll look at Centrifuge. And you add your sample. Okay, and so right now, like I said, Centrifuge only has the one database, and we have not implemented the options that are available in the command line application. So all you can do is select a sample and click run. And now look at the results. Okay, and so just like the other two apps we've added, you have a results table with the taxa and the number of reads that are mapping at each level. You create a dendrogram. Again, there's quite a few taxa found. And sorry. And then also you can create a Chrono Plot, which also has quite a few taxa found. And so these are the different apps. So you'll notice that if they're fairly easy to use, but if you're actually interested in doing a large number of samples and you want to run it using several apps, this actually gets very tedious. I did this for GSP last year, the 2019, where I had 20 or 30 samples that several of whom I wanted to run as a group or do the same exact thing, same exact analysis, and I wanted to do a comparison of them. So what we did is we added the ability for implementing read sets. So a read set is a group of reads that you want to run as a unit. So to do that, you would you would use a utility. Here, let me just read. Under utilities, I think is one place to look for it. Build read sets. And that's very simple. You just add. So you have, for example, I had a bunch of samples I want to run. 808, for instance, is one. And you can use their handy search tool to find them. Okay. And then you give it a and then you click run. And then you've got this group of samples that you can then like run QC on, and then take the output of the QC to run Gotcha2, Centrifuge, Kraken, Kaiju, whichever you want to run. And it makes your life much, much simpler. Okay, so here's some outputs of samples I run using that have been grouped together as read sets. So this is a monosporascus sample, and you'll see that they're grouped into tabs. Let's see, finish loading up. Okay, this one finished first. Okay, so each read is grouped into its own tab. So, and then you can quickly just click each page to find the result for that particular read sample, and so on. So there are 8 samples, so 8 different pages, and you get the same output. If you're comparing them, you probably wouldn't open them all into their own separate tabs, and then you can just click back and forth between them. So that's it for the demo. Let me go back to the let me go back to the PowerPoint. And go to, okay. Okay, and so in summary, there are 2 taxonomic profiling apps that have been released to KBase, Gotcha2 and Kaiju. 2 that are still in beta, Centrifuge and Kraken 2. For pre-processing, if you want to probably run pre-processing first, Trim-O-Matic is the one that's in KBase to remove the low quality reads. And if you're working with a large number of samples, I would use read sets, but that is only available right now in beta for Gotcha2. I will be adding that to the released version. And it makes it much easier if you're running same group of samples with multiple apps. So take home, so there's no one taxonomic profiler to rule them all. So there's trade-offs in speed, accuracy, and the type of analysis you want to do. Gotcha2 has high specificity and sensitivity, but it's really only meant to be used for per sample taxonomic classification. It will not classify every single read, since it is throwing away a bunch of information when it's creating that database. So it's only trying to determine which what OTUs that sample belongs to. Centrifuge and Kraken are a bit faster. Those are meant for per read classification. So if you want to classify every read in your sample, you'd want to use either Centrifuge or Kraken 2. And also, if you have 16S ribosomal RNA, you would want to use Kraken 2. And my suggestion is, if you pick a memorable name for your app, I like Kaiju and Kraken because you can't go wrong with monsters. I'd like to thank Shane Cannon, Paramveer, and Boris for helping us get these apps into the KBase App Store. Alicia has been great in keeping everything organized for SFA. Ben has been great helping me with the webinar. Megan has been wonderful in keeping us on track to get all our tasks done for the SFA. And I also like to thank all the folks at KBase who helped us with the workshop in January. If you want, I think if they're still doing the KBase app SDK workshops and you want to add your app to KBase, I would really recommend attending that, since that gets you helps you really get a good idea for how to get that done. And then I'd like to thank the KBase SFA and Patrick, our PI. So I'll check to see if there's any more questions in the chat. I think I might have answered them all.
Maybe.
Oh, okay. Sorry. How does Gotcha2 deal with complex samples where the likely taxa that don't match reference databases? Well, yeah, of course. If the sample, if the reads aren't in the reference database, then yes, they will not be found. We're trying to keep them up to date. I think the latest one was done in 2020 in the fall, so we'll probably maybe due for another update to the reference database. But I would say it does a pretty good job with complex samples, since it does filter out the redundant reads at different levels, so it performs fairly well with complex samples. And it gives you, if you're looking for, if you just want to know which samples are in your which organisms are in your sample, I think Gotcha2 is a pretty good option. Are there tools for combining the output of multiple twos? No, there aren't. That would be a great thing to have. See, some like we could probably do that for Gotcha and Centrifuge and Kraken too. I'm not sure about some of the others since their outputs are very different. But yes, that would be something I think would be great to add to KBase as a tool. I mean, right now, yeah, each one you kind of have you kind of look at it on their own. But so thank you for that suggestion, and we'll look into adding that. Is there any way to multiply FASTQs at a time? I guess the each sample can have multiple FASTQs. I think probably, but I think using the read sets is a great way to analyze multiple FASTQs at a time. So I'd probably say that we're using read sets. Will these work with meta-transcriptomic data? I don't think so, because they're really, I don't think they're not, there are other tools that are probably better for meta-transcriptomic data. We have one that was developed at LANL by one of my colleagues. It's called FAME. I can put that into the, and that was developed by Magoon, one of our scientists. I can include a link to that if you're interested in, but as of now, I don't think there are any, I'm not aware of any apps in the KBase App Store that handle meta-transcriptomic data. Do any of these work with assembled metagenomes? I'm fast. I think Gotcha2 will work with FASTA files, but not in the KBase app as it exists. I don't think you can use. Actually, I'm not sure. I think it might work with a FASTA file if you uploaded it. So in principle, there's no reason why it can't work with FASTA. But I don't remember if we actually implemented that in KBase to accept a FASTA file. But that's a good question. I can check and get back and add that to the to this document. Is the database with artificial reads publicly available? Yes, it is. I can find that as well. A link to that data set. It's in the NCBI. But I can get a link to that and add it to this document as well. Let's see, I think that's
Excellent. Yeah, I'm seeing a few folks sending me questions directly. If you could drop those in the Q&A document. So one of them is
oh yeah, paired end versus yeah, you can use single end or paired end libraries. So when you're running a read, thank you for reminding me. For read sets, they all have to be of the same sort. If they all either have to be paired end or all have to be single end. If you mix them, that will cause an error because you can't compare the. It can only run all paired end or all single end. That reminded me of one of the it checks that in the app. Gotcha2. I think Kraken only Gotcha2 does not work with 16S data. It's also in the question from the chat, but Kraken does. The Gotcha2 database isn't meant for 16S RNA data.
All right. Thanks so much, Mark. I think
Sure.
yeah, I think that's hit the questions. We'll post this webinar up on YouTube if folks want to revisit it or share it with other people as they're using these tools. And of course, as you're using KBase, if you run into any errors or you have any suggestions, you can use our help board or send us a message and let us know. But yeah, thanks, and everyone, give Mark a round of applause for the webinar and a virtual round of applause. And hopefully, we will see you all next month. And our next webinar, we're going to try to showcase the Enigma SFA. So we're working through all of these collaborative development projects that we've been doing for the past couple of years. So hopefully you find these all very informative, and you'll come around for the next one. We'll be sending around a feedback form if you could fill that out. And otherwise, have a great rest of your day.
Yeah, thank you everybody for attending. I really appreciate it.
Thanks for watching this webinar. For more webinars and tutorials, subscribe to our YouTube channel. You can see announcements for upcoming webinars and recordings for previous webinars on our website at kbase.us/learn. Let us know in the comments what content you'd like to see in future webinars. Be sure to follow us on Twitter at DOE-KBase. And if you have questions or encounter an error when using KBase, please contact the help desk.